पवित्रवतु।
This lecture, you will be introduced with the components of cytoskeleton, the origins of cytoskeleton, what different components exist in eukaryotes, and we'll see in last set of questions whether they exist anywhere else. Okay. Then we will first start with one of the three components of cytoskeleton, that is the microfilaments. We'll see what are their functions, what are they made up of, that's the actin structure and their dynamic properties. And finally, if the time permits, we will see actin assembly and kinetics. Okay, so what is cytoskeleton? Cytoskeleton is a network of protein polymers found in the cytoplasm of the cell. Okay, it is just like as if you have an endoskeleton. What is an endoskeleton to a body is the same way you have cytoskeleton to a cell. Okay, so we see what are the components of cytoskeleton, what elements exist. There are three in particular. There is a fourth class which does not get discussed, which are septins. Okay, we'll not discuss them in the lecture, but there are three types. The first one is the microfilaments, the microtubules, and the intermediate filaments. They are named in such a way that they are based on the size. The microfilaments are the thinnest of the three elements, whereas microtubules are the thickest of the three. Intermediate filaments are in between the size of the thickness of the microfilaments and the microtubules. And down you see the localization of each of this filament by a fluorescence micrograph. Okay, we'll see this shortly in the next slides. Okay, so yes, these are the actin filaments. As you can see, they form assemblies in microvilli. Okay, these are the muscle, the Z disc and the muscle contraction. Okay, we'll come to the structure shortly. So there are some functions of actin filaments. It provides overall shape to the cells. Okay, it links transmembrane proteins to cytoplasmic proteins. They also act in signal transduction. Okay, they play a very important role in forming the contractile ring. So microfilaments we'll be doing little later. We just brief quickly. These are the second type of cytoskeleton elements. These are the microtubules. They form a hollow tube-like structure. Usually, the major role played by microtubules is in motility. So there you see, this is how the microtubule localization is found in the cell. Whereas the second TEM is of a cilia. Okay, the cross section of the cilia, where you see the 9+2 arrangement. Okay, down you see the microtubules play a part where they found spindle fibers. Okay, so these are the microtubules. And lastly, the intermediate filaments. Intermediate filaments, they form rope-like structures. So their major role is to play supporting role. So they provide strength, integrity to the cell. And there are different types of intermediate filaments. Not like actin and microtubules, the microfilaments and the microtubules. Microfilaments are made up of actin monomers, whereas microtubules are made up of alpha beta diamonds, tubulin diamonds. Okay, so at this point we ask ourselves a question whether cytoskeleton is a eukaryotic concept, whether they are only found in eukaryotes or they are found in all different types of cells. Okay, so whether they are found in prokaryotes, yes, they are found in prokaryotes, but for a long time it was assumed that they do not exist in prokaryotes. Obviously there was evidence. Based on the evidence, they looked for these proteins in prokaryotes, but they couldn't find it. But over the past 2 decades, it has been clear that there are analogs of cytoskeleton elements found in prokaryotes as well. And there are 3 cytoskeleton elements that you've seen in eukaryotes. The analogs for actin in prokaryotes is said to be that you have the MREP protein, which is similar to actin. Then you have another protein, the SPAR-N and SOPA. Okay, and the relative protein to tubulin in prokaryotes is found to be as FTSZ or FTSZ. Okay, and for intermediate filaments, it's crescentate. Okay, so the interesting part in this slide is you can see actin, the MREP, is responsible for the maintenance of the cell shape, as you can see in the diagram where this is a Vibrio. It's a comma-shaped bacteria. You see the MREP forms a spring-like structure which holds the whole cell together. Okay, and whereas the PAR M and SOPA are shown to function in chromosome and plasmid segregation, which is very striking because this function is performed by tubulins in case of eukaryotes, but where we see the actin filaments, proteins, analogous protein to actin in prokaryotes do this function. Okay, and same way the functions of the FTSZ is also switched. It plays a very important role in cytokinesis. As you can see, the FTSZ forms a ring-like structure, and when the bacteria divide, so on the onset of division, the FTSZ forms a contractile ring which will pinch off the bacteria. Okay, so again this is very interesting because the functions are opposite, but the structurally the protein is similar to tubulin. Okay, and intermediate filaments, one of the examples is crescentate. Okay, so in general you can say that these proteins they evolved through evolution. Okay, they made polymers. Okay, now over time cells found different uses in different contexts for these proteins. Okay, so these three elements, the cytoskeleton elements, the actin, the tubulin, the intermediate filaments, these are nothing but system of polymers. Okay, they are very important. They give functional consequence to the cell, and they are important for the biology of the cell. Okay, so the next, so obviously, what are the general functions of cytoskeleton? Functions of cytoskeleton are in cell shape. To exemplify this further, we take a striking example of an embryonic stem cell. Okay, embryonic stem cells in culture, they show nice. Okay, they are spherical. Okay, when they differentiate into specific types, okay, they undergo a process called as EMT, that is Epithelial-Mesenchymal Transition. What is this EMT? Where the shape of the cell overall changes to a elongated type of cell. Okay, so this is an epithelial kind of morphology, and after differentiation, in the course of differentiation, you see that the cells have now become elongated. Okay, so this is the EMT. Same you can say for a neuroblast cell, which is a precursor to the neuron. Okay, when it undergoes differentiation, it will make a cell from a spherical structure or morphology. After differentiation, it becomes a thin neuron, a nerve cell, which is the structure of which is very critical in establishing the wiring of the nervous system. Okay, so and most importantly, you see another example of a platelet, where a platelet, it keeps on circulating in a system until, in response to a bleeding or a wound, okay, they get activated, and immediately they reach to the site, and they a non-descript kind of structure, which these platelets are very small, you must have seen in the blood smears. Okay, they become irregular in shape. So how is it that, so the underlying question in all these three examples is, how is it that the cytoskeleton was able to do all this? What properties of cytoskeleton allow them to change shape in such a drastic manner Okay, and so in this case it was a very quick response because wounds, in response to wound, the platelet quickly from a spherical structure, okay, it becomes an elongated or a just to cover, okay, to form clots. Okay, so this is one function of cytoskeleton, is in cell shape, maintaining the cell shape. The next function of cytoskeleton is in internal organization of the cell. Okay, what is this internal organization of the cell? A cell is not just full of a mixture of proteins and enzymes and organelles. It has specific microenvironments within the cell. So there are different regions within the cell which have different biochemical makeup. Okay, you take an example of a neuron. Say in this case, this is the dendrite and an axon. Axon is presynaptic, right? So it releases neurotransmitters, whereas dendrites are postsynaptic. Okay, and they are programmed in such a way that they will receive neurotransmitters. So how is it that just one cell and different regions of just one cell are able to perform different functions? Okay, and that is because of the underlying microenvironments. There is differences in the region. There are regional differences within the cell which allow the cells to perform different functions. Okay, so and the reason they are regional differences is because the proteins they interact with the cytoskeleton elements differently. So there are different cytoskeleton elements which interact with the proteins and change the microenvironment within the cell. Another example of setting the internal organization of the cell would be an epithelium. Okay, this is the basal lamina, and you have the blood. Okay, this is the apical region and the basolateral region. So from here we have the basal. An epithelium of this arm, these epitheliums are, you will have tight junctions here. Yeah, so it is a sealing barrier. We have seen this in previous lectures. That what is seen here is the cell, and this is the gut, the lumen. Okay, so nutrients are taken up by the cell. Okay, at the same time, waste are given out. So again, this stent has become asymmetric in function, and the reason it is able to do that is because there are the cytoskeleton element associated with different regions. The apical region is the microvilli. You will find actin filaments more here. Okay, so the regional differences in this apical and the basolateral regions of this epithelium allow this epithelium to behave and perform different functions. With just one cell, it allows it to different functions. Okay, so this is the internal organization of the cell, and yes, the next slide will introduce you to the picture that you are often used to see. You are often familiar with this type of cell where the nucleus is at the center. Okay, surrounding the nucleus you have the RER, the rough endoplasmic reticulum, and just next to the RER you find the Golgi. Okay, the mitochondria are wherever they are required. So how is it that the cell is able to maintain such organization? It's not just like the nucleus is floating here and there, it's fixed. Mitochondria can be moved where place is required. Okay, so all this is because of this is the internal organization. If you use a drug which will destroy the cytoskeleton, which will inhibit cytoskeleton formation, the Golgi apparatus, for example, colchicine. Colchicine is a molecule which goes and blocks the microtubule formation, assembly. If you add colchicine to a cell, the Golgi just fragments into vesicles. It's no more able to sustain or be close to the nucleus. Okay, so organelle transport, shifting the organelles here and there and making them stay at the proper position is very critical for the biology of any cell, okay, to perform specialized functions. The next slide, and the third function that we have is the force generation, means the cell is able to produce force. What do you understand by force generation? Force generation is nothing but contribution of cytoskeleton, the function of cytoskeleton in motility of the cell. So the first function a cytoskeleton performs is in motility, where you see a fibroblast or a neutrophil pursuing a target, it keeps on rolling, okay, the gliding motility that you see is because of the actin filaments. So actin plays a role in motility. There is another type of motility due to the cilia flagella. Okay, so there the tubulin, another cytoskeleton element, plays a very important role. So motility by cilia and flagella. Okay. The next is chromosome segregation. So next is mitosis. Force is required to pull the sister chromatids towards the opposite pole. Yes, in the anaphase, right? So chromosome segregation, then organelle transport. The same with the example that we have seen, the presynaptic axon needs to secrete neurotransmitters in the synapse. So there should be a continuous transport of vesicles full of neurotransmitters, and they should be exocytosed in the synapse. So organelle transport, vesicle transport, where these are the functions where force is required. Okay. So these are the three general functions of cytoskeleton element. Cytoskeleton. Okay. So in order to understand how cells are able to feature so many beautiful properties, is to understand the underlying cytoskeleton. Okay, and for cytoskeleton to do all this, it has to have two properties. These properties are very important. These two properties seem to be contradicting each other. See, for maintaining the cell shape, for example, it requires strength to withstand the compressive forces. So one property that is required by cytoskeleton is the rigidity. It should be very strong, okay, it should be stable, and should be able to withstand the compressive forces. Okay, in this diagram where we have seen there is a microvilli, okay, this cell adopts a spherical shape because it is the most energetically favorable shape. It requires a huge cost of energy to just push the membrane out and keep it there in that position. So, okay, that is the property of cytoskeleton is, one property is rigidity. Okay, so if we say this is just one property, what will happen if the cytoskeleton is just rigid? The neutrophils, which is pursuing a chemoattractant or a target, when it will start making actin filaments, and the actin filaments are so stable that once they are formed, So the neutrophil will just pause. Okay, so it has to have a property which allows it to be regulated. So there has to be a dynamic instability or intrinsic instability. Okay, so this example is fibroblast and neutrophil, which require chasing of the target and nullifying the target, the bacteria in an immunological response. Okay, so these are the two properties which are important. But the critical question that we ask here is, how is it that the cytoskeleton can be both rigid as well as unstable? Okay, see, this is one of the key questions that we are going to answer in this lecture. Okay, and with this we start with the first cytoskeleton element, that is the microfilament. The microfilament has been seen to assemble different structures, and in short, in other words, you can say this is the localization of the actin system. There is a microfilament. The first case we have already seen, there is the microvilli, where you see a parallel bundle of actin filaments running. Okay, and the second structure is in the cell cortex. Okay, actin filaments are shown to form cross-linking network just below the plasma membrane, and they seem to be associated with the plasma membrane. Okay, to give cell shape and support. The next assembly that you see often is, we have seen in the previous lecture, is the belt desmosome, where the belt desmosome has internally the actin filaments running through, which also provides shape of the cell. The second example is the striking example where the cell which is moving. So the cell, you see, this is the leading edge where the movement of the cell is upwards. The part of the cell which is lagging behind is called the trailing edge. Okay, and a cell which is moving will show filopodia, lamellipodia, and stress fiber formation. These three are very important for maintaining the overall structure while it is motile. Okay, filopodia are just extensions, small projections helping in direction. Lamellipodia, they help the overall cell to form a flat like structure, and stress fibers are contractile actin filaments. Okay, and then these are other different assemblies or localization of microfilaments. So during phagocytosis, it helps in taking up the vesicle and moving endocytic vesicle. It is so far it was seen the tubulins and the motor proteins, they carry the thing, the kinesin and the dyneins, but actin filaments, the microfilaments also play some part in moving endocytic vesicles. And the last, you already know, the contractile ring during cytokinesis. Okay. Now with this we move on to the structure of the core element forming the microfilament. Okay, so actin is a subunit which makes up microfilament. So actin in the cell can exist in a monomeric form, which is often referred to as G-actin or the globular actin. Okay, and can also exist in a polymeric form. See, initially we said this network of protein polymers. Okay, so it can exist in a polymeric form. This polymeric form is often referred to as F-actin. G stands for globular form of actin, and the F stands for the filamentous form of the actin. Okay, and this actin is a 42 kilodalton monomer, the G-actin. Okay, and X-ray crystallographic analysis reveals that the actin monomer, the G-actin, is actually a structure having two lobules or two lobes. So the whole globulin structure is separated by two lobes and a deep cleft. This is the cleft. Okay, these are the lobes. Now, one very important property of F-actin is when it assembles, it has a structural polarity. Okay, so F-actin has a structural polarity which also has a functional consequence, a functional polarity. Okay, so you have a plus end and you have a minus end. So what is this structural polarity? Means if you see this F actin from one end versus another end, both ends look different. Okay. Now, why did F actin have polarity? We have to see. The answer is the G actin monomers are asymmetric. So when they assemble one after the other, making the one end look drastically different from the other end. Okay, and one very important property of actin is it binds to an ATP. So that cleft, the two lobes and a deep cleft, the cleft has an intrinsic ATPase activity. So you have ATP plus Mg plus 2. Okay, a cofactor required for the activity. So that is why. And a very critical question in understanding how actin performs its function is to understand why actin should bind to a nucleotide. Now, why actin binds to ATP? Okay, and in this one, we'll see further in the further slides that what is the consequence of actin? The consequence of actin in formation of the polymer. So yes, you can see the structure. The two lobes have divided into 4 domains, subdomains. The first and the third form the base of the monomer. The second and the fourth one, they form the top one. Okay, the red is the ATP. The tail one is the magnesium. Okay, and the bottom, the first and the third, they form the plus N. Okay, and the top part is the minus N. Yeah, the whole actin. Yes, I forgot to mention that actin is not just for pictorial purposes. Actin is as if a string of beads. It is a helix made of a string of actin filaments. So there is an actin. You can see the star. The filament is the star. It winds over the other filament and forms a helix. So it repeats itself after 14 subunits. Okay, and the total the length is 72 nanometers. And since these are two strings, you see each string is repeating itself at 36 nanometers. So these are the structural properties of actin itself, the G actin and the F actin. Okay, then so the question that we ask is, how do you see this polarity? How do you observe polarity? Conventional fluorescence microscopy has a resolution limit of 200 nanometers, and we did not say the thickness of the actin filament is 7 nanometers. Okay, and the resolution limit of a fluorescence microscopy is 200 nanometers. So we won't be able to make out whether a second actin is present close to each other, okay, next to each other. So it is difficult. So fluorescence microscopy is out of the question. Only with the property of X-ray crystallography, the resolution offered by X-ray crystallography allows us to observe that intrinsic, this polarity, structural polarity. But there is another trick to doing that. We can use an electron microscope and something called as the decoration experiment, okay, to visualize the property, this polarity, the structural polarity, in a much easier way. Where what you do, you use you take myosin. Myosin is an actin binding motor protein. So you take myosin, you digest this myosin using a protein like enzyme or proteases. Okay, and what you get is an S1 subunit. The S1 subunit is, suppose this is the myosin, there are two heads in fact. Okay, so if you cleave myosin using a protease, you get an S1. There are the heavy meromyosin and light meromyosin, but the fact is we want the S1 myosin, which is nothing but the actin binding unit of the myosin. But this S1 will bind specifically to the actin filament. Okay, when you add S1 into a cell, or okay, what happens is it binds to actin at a particular angle. Okay, and when you observe, when you do an EM, okay, you see that one end you see the S1 is sort of they are arranged apart, whereas the another end which seems pointed. So there is another terminology to say, okay, this the barbed end means barb of an arrow, and there is a pointed end. Okay, so the pointed end is the end where you have the minus the polarity, and the plus end is a barbed end. Okay, so these are the small. This is a small experiment which can be very useful in observing the polarity of the actin filament itself. Okay, so okay, so the next is we are going to see something about the actin assembly and the features that actin filaments show in vivo. So to further elaborate, if you are an actin filament and if you want to join this actin filament, okay, as I said, the polarity, there is a structural polarity and it also has a functional polarity. So if you are an actin filament, if you want to join this filament, this is an experiment, okay, where I take this filament in a test tube and I am flooding more actin monomers, the G-actin monomers in that test tube. What I see is the assembly occurs 10 times faster than at the plus end than as at the minus end. Okay, so the plus end is also often referred to as the preferred end for assembly, whereas the minus end is not that preferred end for assembly. Okay, so far we have seen the actin filaments, the decoration experiments, and we have seen which is the preferred end and what the structural polarity gives a functional consequence to the polarity. Okay. Yes, the next is the actin assembly. So, in this slide we are going to discuss about the features that are seen when actin filaments assemble in vivo inside the cell. So, what you see is assembly of actin filaments does not occur spontaneously. Okay, instead it occurs at the membranes. Okay, actin filaments nucleate most frequently at the membranes. How can we demonstrate this? So, it does not occur spontaneously. Assembly occurs at membrane. Okay, so what you can do, we can take a cell, okay, and we can micro-inject a fluorescently labeled actin molecule. Okay. So, and we quickly ask what will happen just after addition of the fluorescently labeled actin molecules. What we see is the fluorescence is prominent at the membrane, which you can also see in this video. Okay, so you can see that the new filament is being formed. These are the osteosarcoma cell lines. You can see at the periphery. You can see the periphery. There is an increase in the fluorescence and it goes away. So the same way, we take a cell, we micro-inject a fluorescently labeled actin molecule, and we quickly observe what happens. We see the fluorescence starts or emerges from the membrane. Okay, so that tells us the assembly occurs at membranes because we have just added the fluorescently labeled actin molecules. The third observation that we see is the barbed ends are associated with membrane. How can we demonstrate this? We can do decoration experiment. We can fix this cell using formaldehyde. Okay, we can add S1 myosin, let it bind, and we do EM. We can see that the S1 sort of decorates the actin structure inside at an angle, and you can see that the barbed ends are all associated with membrane. Okay, and the last and the fourth very important feature that we see in actin assembly is F-actin turns over rapidly in the cells. The turnover means F-actin does not just form and just stop. Okay? The turnover of F-actin is very fast. To demonstrate this, you use fluorescence microscopy techniques. There are two techniques by which you can demonstrate this. One is called as the FRAP analysis and the speckle analysis. So in this video, you can see the earlier video was a FRAP video, okay, and the second is the speckle analysis. So what happens is speckle and FRAP. So we will take this example where FRAP stands for fluorescence recovery after photo bleaching. Okay, so you flood in a lot of fluorescently labeled actin molecules inside the cell. Okay, and let it equilibrate in the sense all those actin, fluorescently labeled actin should be part of the F-actin form. Okay, assemble. What soon you will find that the entire strands will be fluorescing. Okay, then you use a laser to make a very sharp band, as you can see in this video. Okay. Yes, so you can see this band, and you observe this band whether the band recovers its fluorescence. If it does not, so what you do, you using a laser, you bleach an area, okay, and you observe it whether the fluorescence recovers or not. If it does not, means actin is a very stable structure. But in fact, what happens is, you have seen in the video that it moves. So the whole band sort of moves on an escalator. It feels as if, okay, and then it disappears. So it turns over. These molecules are taken assembled inside into the F-actin and they dissociate. So they disassemble at one end and they assemble at one end. Okay. The speckle analysis is a slightly different version of the FRAP, where you don't require laser, but instead what you do is you decrease the amount of fluorescently labeled actin molecules you put inside the cell, such that you get occasionally one or two fluorescently labeled actin molecules taking part in the assembly of the F-actin. Okay, and that gives you a oh, yes, the picture on the right, the video on the right, you see there are lots of speckles. Okay, and these speckles, they seem to go down. It's just like your airport, where you have lights, they keep on moving to show the direction the same way. Okay, so that was a you can see small speckles means individual actin molecules. They are fluorescing and they are moving on the F actin, showing that it is highly dynamic. Okay, so the next property is to understand. See, we have yet to discuss why the assembly does not occur spontaneously. So we have seen the features in vivo, and we need to confirm them in vitro. We can study the actin dynamics, assembly dynamics, the kinetics in vitro in an experiment, where what we do is we isolate the actin, F actin, and for the monomers, the G actin, we can put salt and start the assembly. Okay. So percentage of actin subunits in filaments was as a function of time after the salt has been added. When you see a graph which is peculiar, is shown, there are three phases in the graph. There is a lag phase, then there is an elongation phase, and then there is a plateau or a phase after a point beyond which there will be no growth. So the lag phase, why do you think there is a lag phase? Lag phase tells you something that actin monomers, they are difficult to assemble initially. Okay, so what happens is, you see that the actin subunits, they occasionally come together and form a dimer. So monomeric units, they form dimers, and the dimers are not stable. So they, as quickly as they are formed, they are dissociated or they disassemble. But occasionally there is a third actin which comes and collides and makes a trimer, or it is also called polygon, more than three. So once that happens, it acts as a seed. It acts as a seed for starting the assembly of the actin filaments. So then you see the elongation phase, where the actin concentration in the test tube, there is a net growth in the filament. So the filament keeps on growing until a point is reached after which there is no net assembly. That point is called as the steady state. Okay, and the concentration of actin monomers, it required for maintaining the steady state, is called as a critical concentration of actin filaments. Okay, actin monomers. So the concentration of monomers at steady state is C sub C, critical concentration. So only at critical concentration of actin monomers, you will find that the filament is in equilibrium with the monomers in the suspension, okay, or in the cell. So there are three phases. So you can say that the rate limiting step in this graph is the nucleation phase. That is because you require that polygonal to form, and that after that formation, the filament will elongate beyond till the point you reach a steady state. Okay. So what if you precede that polygon at the start of the experiment? Okay, instead of having G actin, okay, you seed that polymer and put it at the beginning of the reaction. What you see, you don't see a lag phase, and you see a elongation and then the steady state. So that tells you that that step is a rate limiting step. So the nucleation step is a rate limiting step. So that is consistent with the point that actin does not occur spontaneously. The assembly of actin does not occur spontaneously. Okay, so this is the assembly kinetics. Then we move on to the very important part, the answer to a question that we asked: why actin should bind to ATP? So the significance of ATP required. See, cells are investing a lot of ATP in making microfilaments, in making cytoskeleton. Okay, which is rigid as well as unstable. So why does cell invest a lot of ATP in building microfilaments? There are two answers to that. First, the ATP acts as a chemical switch. See, in the diagram that I showed, the cleft was bound to ATP. The ATP actin likes to assemble. It wants to form polymers. Okay, but as it assembles, the ATP in the F actin, okay, is hydrolyzed to ADP plus PI, and that causes, so the ADP actin now does not like to assemble. It likes to disassemble. So addition of ATP makes the filament unstable. So that is one reason why cells are investing a lot of ATP. The second is to allow two ends of F actin to behave independent to each other. Okay. So for that, we will compare and we will see a hypothetical situation, a graph where the system, the cytoskeleton system, does not have any ATP. How does the two ends behave then? Okay. So as I said, the steady state, that is known as assembly. So, and the plus end is the preferred end. So the assembly occurs 10 times faster at the plus end as compared to the minus end. So we will see a quick graph. We will see a quick graph. So about the 0 line, you have the plus edge of assembly, means the filament is forming, okay, and below you have disassembly occurring. And as you go towards the right, you see the actin concentration increase. So this is the actin concentration, and this is a case where there is no ATP. Okay, so you see that the plus end. This is the plus end. Since the assembly at plus end occurs faster than the minus end, so it will quickly, as the concentration increases, it will quickly assemble. Whereas the minus end, the assembly that occurs at the lesser speed, will have a graph like this. Okay, and in this graph, there is a point where the two lines they intersect. This is the C sub C, the critical concentration at which there is no net assembly or disassembly. Okay, so this is the case where there is no ATP, and in a real situation where there is ATP, so you have a graph. Now, addition of ATP, as I said, it makes the filament unstable and allows the two ends to behave independently of each other. Okay, so it is related to the polarity of the filament. Okay, so the plus end, as the assembly is very fast, will assemble. At the plus end there will be assembly, and the minus end now does not require to be dependent because it is independent of each other. As it grows very slowly, it has a graph like this. Okay, so it grows much slow at a slower rate. In this graph, the plus line interacts with the concentration of actin. This is called as a C sub C plus, and this is called as a C sub C minus, where there are two critical concentrations now. Okay, a critical concentration for the plus end and a critical concentration for the negative end or the minus end. Okay, this you cannot find in the system where there is no ATP. So the ATP allows two ends to behave independently and thereby have two different critical concentrations. And it is an obvious fact that since the assembly at plus end occurs very fast, the critical concentration required, okay, would be very low. Whereas at the minus end, the assembly is not that fast. So the critical concentration is very high. So this has been measured for the cell. The C sub C plus is around 0.12 micromolars. C sub C minus is around 0.6 micromolars. Okay, and yes, one more point. There is a point in between this C sub C plus and C sub C minus. Okay, where you see the assembly and the disassembly at a particular concentration are the same. The rate of assembly at the plus and the rate of disassembly at the minus end is same. So at this concentration, the plus end is growing and the minus end is disassembly at this point. So the rate of assembly at the plus end is the same as the rate of disassembly at the minus end. Okay, this is called as a CS, a special critical concentration, where a phenomenon seen in actin filaments happens. That is, the treadmill occurs. What is treadmill? We will see in the next slide. Okay, so this is the range of treadmilling zone. This is also called the treadmilling zone. We will see. So is it understood? And the critical concentration, the C sub S, the C sub C is okay. So the next slide. Yeah, the ATP and the intrinsic polarity is the same. This is the situation that is seen in the cell. You have two graphs. The T stands for the filament where you have the ATP actin getting inside. See, the adding and the removal unit, the assembly unit and the disassembly unit are different now. In the case number 1, where there was a hypothetical situation, no ATP, the assembly units and the disassembly units were the same because ATP was not involved. But when ATP, you add ATP in the system, the monomers, the G actin which is getting inside the filament is in an ATP form, whereas the one which is getting out is in the ADP form. So this is also one of the reasons what ATP contributes to the filament, the instability of the filaments with its intrinsic. So ATP and intrinsic polarity, this is a treadmilling range when all these concentrations of actin will find the plus end will be growing and the minus end will be disassembling. So there is no net growth of the filament. The length of the filament remains the same. Okay, and on the right hand side you see a light imaging where right from the, so this filament, the green thing, is in equilibrium with the G actin in the cell which are labeled. Okay, and as you see the filament length remains sort of same or similar, and it appears to move, right? You can see at the 98th second it moves ahead. So this is what is called as a treadmill. Okay, so this is a treadmill. Okay, yes, we have the values. The C sub C is 0.12 and 0.6. Yes, the rate of addition is 12 micromolars per second. Okay, so as I said, the additional assembly units are the ATP actin, and then since the rate of addition at the plus end is much faster, you find a stretch of filament which is still in the ATP state, and then in the middle region you find the hydrolysis. So the speed of adding the ATP actin is much faster than the hydrolysis, so there is a lag in the hydrolysis. Whereas that is not the case in case of the minus end. The minus end, the assembly is not that fast. So the rate of hydrolysis is faster at the minus end than the rate of assembly. Okay. The second diagram tells you that if an actin, ATP actin goes inside to the plus end, now this is the state where you are treadmill. So it goes inside, and the length of the filament remains the same, whereas the actin just flux out from the filament. So new and new actin molecules, they come inside the cell, okay, and they are disassembled at the minus end, as if you have a pipe and the water goes, the length of the pipe remains the same, the water goes inside out, the same, similarly. Okay, so this is it about the actin dynamics, the assembly and the dynamics. The next lecture will come with new question. That, see, there is a question we can think upon in the weekend, that the intracellular concentration of actin is around 200 micromolars, which is 100 times more than the critical concentration of even the -10. So how is it that the cell is not continuously making actin filaments? Okay. So in the next lecture we'll think about this question, and we'll discuss more about the actin binding proteins, the role, and ultimately the motor proteins. Thank you.